having the session from Professor Shailendra Sharma now. He's from Chaudhary Charan Singh University, Meerut.
Thank you. Desktop 101. Just a moment, please.
Sir, can you please share your screen? It will show directly.
Yeah, I'm doing that. Just please wait. Yes.
Yes.
Are they visible? Slides are visible?
You're audible, sir, and your slides are visible too.
Thank you, ma'am. So, thanks for organizers for this Agri-Vision conference. Unfortunately, I could not attend this program in physical mode, as I told that my mother is very ill, and I am also suffering from severe cold. Anyway, so once again, thanks for the organizers for giving me this opportunity and accepting my request to deliver this lecture online now. So my title is Dissecting Genetic Resistance of Wheat towards Plant-Parasitic Nematodes. So before starting, I would like to give brief introduction about these plant-parasitic nematodes. So these are tiny, complex, unsegmented roundworms and are anatomically differentiated for feeding, locomotion, digestion, etc. These species are transparent, L-shaped. So plant-parasitic nematodes are very economically important. They have a styleted mouth, they puncture cells with the help of this stylet, and more than 2,000 species of plant-parasitic nematodes are known. So they are basically known as the hidden enemies of the farmers, because the symptoms caused due to this damage caused by these plant-parasitic nematodes are even confused with the nutritional deficiency in the plants. So the most common damaging PPNs are the root knot nematodes, cyst nematodes, and root lesion nematodes. So basically, I am going to talk about two nematodes on which in my lab we are working on two nematodes, and basically we had a DBT-funded project, a major research project. One nematode is Heteroderma avena, another is Petilenchus horni. One caused the well-known Molia disease of a small cereal grain species like wheat and barley. Other cause root necrosis. So when we talk about this Heteroderma avena, it causes a serious economic problem known as Molia disease of wheat. It can cause yield losses up to 4.3 to 100% in different countries, from Pakistan, India, Morocco, Saudi Arabia, and especially in India, when you talk about the Western Haryana, Rajasthan especially, so we have this severe problem of this Heteroderma avena. And there are no significant genetic studies on Heteroderma avena. So when we talk about the management, we can go for crop rotation, sanitation, tillage, biofumigation, nematicides. But the most eco-friendly and sustainable management is the development of the resistance varieties. So I'm going to talk about the genetic research that we conducted on these nematodes. So in my lower portion, you can see these are patches. So these are the empty patches in the wheat field. So these are the due to nematode infestations of the roots. Video.
Plans to develop.
I just want to show one video.
Already at this early stage, the root is well developed. In many soils, dark brown, thick-walled cysts lie hidden. Many of them have lain dormant here for years. These cysts often contain hundreds of eggs. Each egg contains an infectious juvenile of the nematode Heteroderma. Substances released by the roots of the young sugar beet plants stimulate hatching of the juvenile nematodes. The nematodes break through their eggshells and then emerge from the cysts. As soon as an infectious juvenile has reached a sugar beet root, It uses its stylet to penetrate into one of the cells of the outer root layer. The roots of sugar beets are suitable hosts for this nematode species. Using the strength of its muscles, the nematode migrates through the root tissue. Supported by numerous cell wall digesting enzymes that are secreted through the nematode stylet, the juvenile advances from one cell to the next, though without feeding.
I think we don't need to show. So I just gave I gave the video presentation for the heterodera shecti. For the heterodera avena is also little more same. So see, these are the pictures from our lab experiments. So it's showing life cycle of heterodera avena. So on the right hand side, you can show the number of cysts. And from this cyst, as shown in the video, after hatches, juveniles come out, and these juveniles move into the soil, and then they through the stylet, they infect the roots, they spread in the roots, and they form syncytium in the third stage juvenile, and later on the females become dead, and later it form, there's some tannization, and there's a formation of the brown cyst on the roots. So this is the whole protocol that we follow in our lab. This is not a very expensive experiment. It shows how we collect the cyst. So we have collaboration with the CCS University, Hisar Agriculture University, where we collect the infested soil from that region, and then we collect the cyst from the infested soil, and this is a protocol that shows how we we infect the plants, and later on we collect the cyst and we count the cysts. So all the experiments were performed in the controlled conditions. So we have controlled conditions, humidity, temperature, etc. So around 60-70 days after sowing, we can find these type of white cysts. So when they mature, they become brown, and inside these we can find lot of eggs containing juveniles. The first experiment was published in Molecular Genetics and Genomics Journal. So it was based on Jiva's genome-wide association study. The title was Genome-wide association scan of cereal cyst nematode resistance in Indian wheat germ plants. So we used 143 wheat genotypes collected from different pen regions of India. We obtained this material from National Bureau of Plant Genetic Resources, New Delhi, NBPGR. So basically it's showing, so as I told, it's a genetic study, so it's showing SNP. So we used SNP molecular markers for this Jiva's study. So basically it's a circular Manhattan plot. It's showing the highly significant MTAs. So MTAs are basically regions, we can say that they are the potential regions or potential genic regions which can tell about the potential candidate genes involved in the resistance against these nematodes. On the right hand side, you can find the chromosomal location of these MTAs. So this study was published in Molecular Genetics and Genomics. Another study was published in the Scientific Reports by Chaudhary et al. This is the identification of genomic regions associated with the cereal cyst nematode resistance in spring and winter wheat. So we have this winter wheat and spring wheat panel from Institute of Plant Genetics Research, IPK Gattersleben, Germany. Again, this is the Manhattan plot, and these are the potential MTAs. These are the chromosomal locations of these MTAs. And the good thing is that we confirm that these MTAs are real from the previously conducted studies. So some of the MTAs that we found in our study were already co-localized with the already discovered major genes of QTLs. So now we are focusing on these regions. So this is a chromosomal map showing MTAs detected in the present study. This is the expression profile of the candidate genes. So after doing this mapping, we concentrate on the finding of the potential candidate genes in order to find the potential genes. So we have different genes like WD40, F-box gene, NVCLR proteins, etc. This is also very another interesting study conducted by Pundir et al., also published in the scientific reports. It's based on the QTL mapping for resistance against serial cyst nematodes in wheat. So the just without, I would like to go to. So as you show, this is a major QTL region that we found, and this QTL. The good thing is that this QTL was already reported few years back, and this shows that our experiments were accurate and precise, as we also reached on the same location that was reported earlier. Basically, we use synthetic opata mapping population, DH lines, double haplod lines for this experiment, and now we are focusing to narrow down this region. So this chromosome was this chromosome was located on the 2D region, and we are now interested in to narrow down this region. So we made crosses between these DH lines. This is another study, also conducted by Pundir et al., published in Genetic Resources and Crop Evolution, Grace. So these pink threads are the stained nematodes. So in order to confirm that whether nematodes there is infection in the root is successful or not, what we do, we stain the infected roots. So after this staining shows different stages. For example, this one is the resistant line M6, and this is the susceptible line Opata. So in the resistance line at 1 dpi, the number of nematodes is almost same like in the susceptible line. But as the time goes, so around 8 dpi, we can found that in the M6 resistant line, you can find only the juveniles, like J2 stage. But in the Opata susceptible line, it is almost conversion of the syncytium. So you can find even J4 stages. So we can see that in the resistant lines, there is a slowing of the progression towards the maturity of the nematodes. Again, we also performed a meta-QTL study based on the previous reported QTLs, and we found an interesting reason on the 2A chromosome. Now currently we are also performing some transcriptomic analysis of the infected root samples. Again, these are the infected roots. So we performed these experiments in replications. So this work is going on, not published yet. So in the previous slides, we used M6 and Opata that we used in our QTL study. But in the another study based on transcriptomic analysis, we used Raj MR1. So Raj MR1 is the only release variety from India which is had to have an infection. So we are using this variety for this transcriptomic analysis. So the another nematode that we are working on the Patelenchus thorni. There are not so many genetic diseases available, especially in wheat. It can cause up to 62 dies, right? Yugoslavia, Morocco. Again, the story is the development of the resistance varieties. So in the previous nematode heterodyrivina, there is a formation of the cyst, but in this case, there is no formation of cyst. So it's a you can say it's a moving nematodes, migratory nematodes. So in this cartoon shows the penetration of the root by the nematodes. Again, there's a formation of the juveniles in the egg development, and first molting J2Hs from the egg, then to second molting, third molting J3, J4, and there's a differentiation of the adults, female and male, and so on. So this infection, the difficulty with this nematode is how we multiply. So initially we have this collaboration with the IPR Kanpur, and we face lot of problem in multiplying these nematodes. We use carrot disc to multiply the nematodes, but now we are successful. So it takes about 3-4 months to get these nematodes and perform the infection. So this is a carrot callus, and this callus contains lots of nematodes. And again the process is same, that we collect the nematodes, and in the controlled conditions we provide the nematodes to the wheat root plants, and then finally we collect the nematodes. In previous case we collected the cyst, but in this case we directly collect the nematodes, and then we count the nematodes, and then we try to associate with the already available genetic markers. In our case it's SNP, single nucleotide polymorphism, genotyping data. So it just shows there is a tissue paper. It contains lot of nematodes which are visible only in the microscope. You can see it's a lot of hundreds of nematodes are there. So this study was also published in the scientific reports. This is a circular Manhattan plot. And again we find some interesting regions that we are currently working on. Again, in the same case also we are performing some transcriptomic analysis for this nematode in the resistant and susceptible genotypes. This work is going on. And the other thing is that now we also developed in the last 5 years, we also developed some mapping population. So what we did, in the first case we used this only released variety from India, Rajamar1, and we crossed with a susceptible variety, WH147. Now it's in F6 stage. And the other one is the WH147, it's a reverse. And the other parent is Rajamar1. So it's a reciprocal cross. So we performed this experiment at Dalang Maidan, Himachal Pradesh, Wellington, spelling, sorry for the wrong spelling, and also at, of course, at Meerut. Another cross. So these are the publications from my lab in recent 4 years. So 3 publications from scientific reports. One is in molecular genetics and genomics, another is in BMC plant biology, another is in genetic resource and crop evolution. Now we are working on the development of the Cas markers. We want to validate the MTAs. We want to develop some user-friendly markers. So now our next goal is to finalize our transcriptomic study and the development of the diagnostic Cas markers. So this is our group. My mentors, Professor P.K. Gupta, Professor Balian, myself, my better half, Dr. Shweta Sharma. These are my students, Deepti Chasduvedi, Deepak, Saksham, Vikas Kumar Singh. And we have collaboration from the Scotland Rural College, Dr. Rajiv Sharma, United Kingdom. And I have collaboration with the IPK Garters Lab in Germany, Dr. Marian Roder, Andreas Bonner. And from NBVJ New Delhi, I'm thankful to Dr. Sandeep Kumar. And that's from CCSU Haryana. I have a very good collaboration with the late Dr. Bansa Singh, Dr. RS Kavar, sorry. And from the IIPR Kanpur, we have Dr. Bansa Singh. So I am thankful to all collaborators and my students and my mentors for making this research successful. And I think very few groups are working in India on these plant parasitic nematodes, especially towards genetic studies. So once again, thanks a lot. Thanks for your patience and listening to me. My voice is cracking due to severe cold. And once again, thanks for the organizers who gave me this opportunity to attend this conference.
Thank you so much, sir. Ma'am, would you like to ask any question?
I think it's not so cold there, Katak.
Yeah, okay.
It's a very severe cold there.
Please take care of yourself.
Thank
sir.
you, Dr. Sharma, for your extensive study in nematode resistance. And
Welcome.
that my question is that you have gone for that pedigree method for this Meerut somewhere I have seen. And then what is the purpose of it? You want to develop the real or something?
Yeah, of course, ma'am. So basically, we are working for the QTL. We have to perform some QTL studies, later on we want to just narrow down the QTL region. So actually the thing is that besides this nematode study, we have some other traits also in our mind. So we try to select the most contrasting parents, not only for the nematode resistance, but the parents which are showing contrasting traits for other traits. So we selected these parents, especially Rajamar1, another contrasting parent. Yes, for the mainly for the QTL region. And in the case of in this DH population, we have already made processes between the DH population to narrow down the region.
Then have you developed some marker for this thing? You have not,
Ma'am,
your future studies to go for fine mapping?
yeah. That's why I told that we are now focusing, we are trying to establish this caste system in our lab.
Yeah. Then,
This
otherwise you can introduce this QTL and you can validate your this resistance in further domain.
yes, of course. The ultimate goal is that.
Yeah.
Some high
Okay.
yielding varieties, our target is that.
Okay. Thank you, Dr. Sharma, for your nice
Welcome.
presentation.